The androecium, consisting of stamens, is the male reproductive part of a flower. A typical stamen consists of a long stalk called the filament and the anther, which is the fertile portion of the stamen. An anther consists of two lobes which are connected by a sterile tissue called the connective tissue. Each anther lobe consists of two chambers called the microsporangia. The singular is microsporangium, also called the pollen chambers. Thus, a typical anther consists of four roughly spherical microsporangia. Such an anther is said to be dithecous. Now let's look at the transverse section of the anther. Four wall layers surround the microsporangia of the anther. Of these, the outermost layer is the epidermis. Beneath the epidermis is the endothecium, which forms the dry coat of the mature anther. Beneath the endothecium lie 1 to 3 middle layer cells. These cells protect the pollen grains and help in their release. Beneath the middle layer of cells lies the tapetum, which provides nourishment to the developing pollen grains. When the anther is young, the microsporangium encloses cells of the sporogenous tissue. Each cell in the sporogenous tissue has the potential to develop into a pollen grain. As the anther matures, the sporogenous cells in the microsporangium divide mitotically to produce the microspore mother cells. Each microspore mother cell undergoes meiosis to give 4 haploid nuclei that are arranged in a tetrahedron. The haploid nuclei are soon surrounded by cell walls and form the microspore tetrad. The process of formation of microspores from a microspore mother cell is called Microsporogenesis. The haploid cells of the microspore tetrad dissociate and develop into pollen grains. The pollen grain represents the male gametophyte in the life cycle of angiosperms and gymnosperms. At maturity, the partition walls between the neighboring microsporangia break to form the pollen sacs. When the pollen sacs are entirely filled with pollen grains, the anther bursts open to release the pollen grains. Did you know that pollen grains collected by bees are consumed as food supplements? Pollen grains are considered to be high in nutrition and play a beneficial role in human metabolism. However, in many individuals, inhaling pollen grains causes severe allergies. A pollen grain contains a single haploid nucleus. A pollen grain contains a single haploid nucleus. A pollen grain is usually spherical and has a diameter of 0.025 to 0.125 millimeters. However, pollen grains come in different sizes. Out of these, the small pollen grains are well suited for wind pollination. A pollen grain shows a tiny mass of protoplast surrounded by a double-layered spore wall. The outer layer of the wall is called the exene. It is made up of sporopollenin, which is one of the most resistant biomolecules known in nature. It can withstand high temperatures and enzyme degradation. The exene shows the presence of spines or warts and is uniquely sculpted. At some places, the exene of the pollen grain remains unthickened and lacks sporopollenin. These are the germ pores. Pollen grains from different plant species show uniquely sculpted exenes under a microscope. Differently sculpted X scenes of pollen grains are of great taxonomic value. The study of pollen grains is called palynology. It is useful in paleontology, archaeology, and forensics. The inner wall of the pollen grain is called the intine. The intine is thin and is made up of cellulose. The pollen grains begin to germinate inside the anther lobe. The nucleus of the pollen grain divides mitotically to form two nuclei, the generative nucleus and the vegetative nucleus, also called the tube nucleus. A membrane surrounds the generative nucleus to form the generative cell. A mature pollen grain thus consists of two cells, the large vegetative cell, or tube cell, and the small generative cell, which floats in the cytoplasm of the tube cell. In most angiosperms, the pollen grain is shed from the anther in its two-celled stage. The released pollen grain must land on the stigma of a flower within a limited time frame in order to carry out fertilization. Pollen grain viability is a measure of the percentage of pollen grains that undergo germination. After the pollen grain lands on the stigma of the flower, it ruptures at a germ pore, and the tube cell extends to form a tubular outgrowth called the pollen tube. Both the tube and the generative nuclei enter the pollen tube. The tube nucleus lies at the tip of the growing pollen tube. Inside the pollen tube, the generative nucleus divides by mitosis to form two male gametes. On reaching the tip of the ovule, the tube nucleus degenerates and the male gametes enter the female gametophyte for the process of fertilization.